Right down that quickly. Thank you for making me feel like I'm hurt. Okay. Here we are. Quick business. Office hours are 11 to 12:30. I'm sorry the syllabus is wrong. I haven't had a chance to update the syllabus and re-upload it. But I'm announcing it here and on the recording because you weren't here. So you guys know that what's on the syllabus is wrong, what's on the big poster outside the bio office, and what's on my door is correct. 11 to 12:30. If you need me and you can't get there, just email me and we'll work something out. Exam 1 is coming right up. Someone reminded me in recitation today. Yikes. Okay, 9:30. It's not right up, but it's getting there. Okay, there was an error on the quiz. How embarrassing for me. The question was, why don't cytosolic proteins contain disulfide bonds? And I wrote a reducing environment such as that found in the ER is required. What I really should have written is that a reducing environment such as that found in the cytosol is going to inhibit disulfide bond formation. So, sorry, I'm going to go through all 90 of your quizzes and check to see if you deserve points. If you deserve points, it would be because you didn't answer something like, there are some things that are obviously wrong. If it's obviously wrong, sorry. But if it's, I'm going to see what I can do to make this fair. But these things happen, and thank you very much for letting me know. Okay, any questions about this? And as you see when we talk about this today, you're going to be like, Dr. Ehrlich, how did you mess that up? But whatever. What are you going to do? Okay, so far this is what we've done. It's a lot of words. We've spent a lot of time, we spent about 2 lectures on the whole getting from mRNA to protein and what needs to happen in order to get protein and ways in which you can get different proteins from the same gene and different proteins with different functions. And that's what most of this up until about starting with protein folding, up until there, that's what that is. And so as you start to study, I want you to go through this list. This list is going to constantly be changing. So don't, because I only have so much space on one slide, so I can start consolidating into major ideas. So as you prepare, keep a running total of what's on these lists, and just make sure you know all those things for the exam. Okay? So last time we kind of spoke about protein folding and ubiquitin, and we started to talk about protein import into the ER. Because after you have a protein, so it's great, we have all this stuff that needs to happen to get a protein, but after you have a protein, so what? It needs to go somewhere to do what it needs to do. And the first one we're starting with is a secretory pathway, and that's why we've been talking about imported proteins into the ER, and all the SRP and SRP receptor and that whole process. Then we start to talk about integral membrane proteins. These are these proteins with the transmembrane domains that many of them are adjustment to the cell surface. Others, they might be membrane proteins that work in the Golgi or in the lysosome. But those all make their way through the secretory pathway. And I started talking a lot about protein topology. How topology is maintained from the ER all the way to the Golgi, plasma membrane, wherever it needs to go. And that's an important concept to understand. And so that's where we're going to pick up today, where we left off. Membrane proteins, yay! I spent a lot of time, I'm realizing, I was going over my slides for today, and I was like, I spent a lot of time on membrane proteins. I think I do that not so much because, a third of your exam will be on membrane proteins, but more because I feel like I want people to understand it, and it can be complicated sometimes because there are so many different ways proteins can go into the membrane. So here we go. So we were talking about these signal sequences, and these start transfer sequences, and these stop transfer sequences, and all these different ways that the transmembrane domain makes it into the ER membrane. And, well, before I go into this, just to kind of wake ourselves up and get back to cell biology, why would you want to put a protein into the ER membrane. What is the purpose of a protein going into the ER membrane? I told you that some of you might think this is obvious, but I want to make sure everyone in this room understands the purpose of a protein going into the ER membrane. And again, I'm sad that I forgot to bring a class list. I need to start calling on people who I don't hear from. Yes?
I was going to say transport of materials across the membrane.
Transport of materials across the membrane. Okay, a protein might end up in the membrane because it does need to transport materials across the membrane. Yes, but what is the, that's not wrong, but that's not what I'm looking for. Yes?
It can either work as a receptor for other proteins to get inside the cell or out of the cell.
Okay, so it can work as a receptor for other proteins to get inside the cell, out of the cell. So from the ER? Why is it going to the ER membrane? Yes.
Oh no. Modifications to make sure it's properly folded.
For modifications to make sure it's properly folded? No, you're not too far off. I'm just trying to get more words out of you. She's like, whoa. You're okay. Okay, so what I'm trying to get out of you guys is why are we putting membrane proteins into the ER membrane? Yes.
So topology is anything?
Okay, so that's a means to an end or an end to a means, but not quite. Topology needs to be maintained, but that's not a reason, right? Yes?
To ultimately be expressed in the plasma membrane?
Right, that's it. Sometimes I just want a very simple answer, and you guys are giving me all these great details. But yes, these proteins are eventually going to be on the plasma membrane, on the cell surface. Okay? That's how they get there. They start in the ER, they make their way to the cell surface. Yes, topology needs to be maintained, so the part that needs a signal is going to stay on the cytosolic side, and the part that needs to bind to some whatever is going to be on the outside. Yes. Sometimes there are proteins that form channels, and those same proteins might make it up to the plasma membrane cell surface, or don't forget, there are proteins that might form channels in the lysosome, such as proton pumps. So you're right too, and I just wanted more of you. Anyone want to add to this? Is anyone having trouble with the concept of the fact that all those receptors on a cell surface start in the ER? Everyone gets that? Makes sense? Okay. Did everyone already know that before they came here today? Or did I teach you something? I hope I taught you something. Okay. Thank you. Okay. Now let's move on. So now we're still talking about how all these proteins get in and why topology is important and all this stuff. So you have here your ribosome translating an RNA, and in this case it has this internal signal sequence, right? And so it translates, and there's this internal sequence that comes out of the ribosome, and SRP sees it and binds to it, and it makes its way over to the ER, and it binds to the SRP receptor, and it goes in and it interacts with the translocon, and that internal signal sequence also it acts as a transmembrane domain. And notice it's not cleaved, and so the amino terminus of the protein stays on the cytosolic side. The ribosome continues translating the protein down, and the carboxy terminus ends up on the luminal side of the membrane, of the ER membrane. So which part, the amino terminus or the carboxy terminus, is going to be outside of the cell once it makes to the plasma membrane? Someone raise your hand. Someone I've never heard of before. All you have to say is amino or carboxy. Which one is going to be on the outside? Let's see if I can remember names. Yes? Amino? Let's see. Amino terminus is on the cytosolic side. Yep. I want to know what's on the outside of the cell once it makes it to the top. All the way to the plasma membrane. I hear you too much. I mean, I have to ignore people I hear all the time. So it's nothing personal. Who else? You're wondering why, right? I don't get it. We get it. Come on, guys. Yes.
Boxes to transport over, it's gonna be on the outside, sides, all sides of the state towards the south.
Right. Right. The carboxy side will be on the outside of the cell, and the amino side, if the amino side's in the cytosolic side, it will remain in the cytosolic side because the topology is maintained all the way throughout transport from the ER to the Golgi to the cell surface. And so you can think of the ER lumen as being the equivalent of extracellular. Well, we can draw it if you guys want. So I'm going to go through a few more examples, and I'll always ask you guys which side is on the outside, and you'll get it. Okay. So remember, we talked a little bit about these plots here that show the hydrophobic domain, so you can figure out where your transmembrane domain might be. So what might this protein look like? Where would you find, so you've got a signal sequence here, and you've got this stop transfer sequence, this high level of hydrophobicity right here. So on this protein, what part would be on the inside of the membrane, and what part would be on the extracellular domain? Did I see your hand going up?
Well, I was wondering if you could explain again how the stop transfer sequence.
So stop transfer sequence is many times acting as your transmembrane domain. So you have the translation. So the question is, can you explain the stop transfer sequence again? So you'll have translation of your protein be coming out of your ribosome. My mouth isn't working today. You guys have those days where your language is not your friend. You'll have translation, and then you'll hit the stop transfer sequence, and it'll literally stop translating for a minute, and that will be your transmembrane domain, and it will move into the membrane there, and then it'll continue translating, depending on. But see, so as you'll see as we go through this, it can be complicated because sometimes you'll have these signal sequences that will act as transmembrane domain / stop transfer sequences. Sometimes with these multi-pass transmembrane proteins, you'll have a start transfer, stop transfer, start transfer, stop transfer. Those are all usually transmembrane domain. You do not need to memorize it all. You need to just understand the concept and then understand that the topology is maintained all the way. So, do you guys want to take a minute, talk to your friends, and try to guess which side might be in the cell? And sometimes what you might need to do is do a little drawing on a piece of paper in front of you, and just kind of guess, because you know your signal sequence is going to come in, it's going to bind to your translocon, and then you're going to have further translation, and then you're going to hit your stop transfer sequence, and it should move into the membrane.
As you're synthesizing or your translation and stop transfer sequence, What's the trans number?
And then restarting cooking. So, like, for example, this is dipping down here. Yeah,
So, Alright, so basically you're going to have a protein.
the sequence of the protein that goes through stuff.
Okay, I just had a question that made me realize I didn't yet introduce the concept of domains to you guys. That's because I usually wait until signaling, because then it really makes a lot of sense. But I'm going to do that right now because maybe that makes sense. Because I'm getting like, what do you mean by this? So, it really just means the part of the protein that does something. So in this case, you have there's your protein, there's your membrane. Remember, this is your lipid bilayer. Does it really look like that? Lipid bilayer. This is your extracellular, and this is your cytosolic. Extracellular domain. This part of this protein is outside of the cell. This cytosolic or cytoplasmic tail. How about that? Who's heard of a cytoplasmic tail? This part of it, that domain is cytoplasmic. This part is your transmembrane domain because it's going trans through the membrane, across the membrane. And so when we talk about proteins, we often talk about different domains of the proteins because different parts of the proteins have different functions. And so as we get into signaling, we will talk even more about it. But in this case where I'm asking you to find, I'm saying, oh, well, this is likely going to be the transmembrane domain because it's very hydrophobic. Because you remember when I introduced this concept to you guys, I said runs of 20 to 30 amino acids that are very hydrophobic tend to be transmembrane domains. And stop transfer sequences tend to be transmembrane domains. Okay? So, who actually stopped and tried to draw out what this might look like? Anyone? No? You guys just chit chat and wait for me to, yeah.
Okay, I was really lost. Like, I can describe what it is, but I wouldn't know how to draw it.
Well, I mean, I'm not telling you to give me an exact. So I'm kind of thinking like, well, if you've got your, I don't know, I'm going to try it. Okay, so there's your translocon, right? And so you're going to have your ribosome come along. There's your signal sequence with your SRP. And so it's going to bring it over here. So your signal sequence will end up binding here. And then you'll have that, right? That's how it'll start. We all agree?
Yeah.
And so then you're going to have more And then eventually you'll have a part that'll be a stop transfer sequence, right? And that should bind there. And then so eventually this will end up being cleaved. But then there will be a hell of a lot more being translated. So then you should end up, I'm thinking, with this. And that should be out, and then this translator will go back in like that when it's done. I don't know, let's see. I'll put up,
Yeah, because the bigger part, because like.
so you're gonna have, is that what I drew? So there's your signal sequence, which is gonna bind, and then your ribosome would be up here. Here's your stop transfer sequence, which is gonna interact also. And so then your signal peptidase cuts that off, that drops down. Your amino terminus drops down. Then you're gonna have further translation, leaving your carboxy terminus up top. How are you supposed to know this? Don't worry about it. Don't worry about it. It was an exercise to see if you can think about it. Because in all reality, it could possibly go either way when you've just got one transmembrane domain, and it has to do with the amino acids on either side. But the key is you're able to recognize your signal sequence and you're able to see your stop transfer sequence. So like if I put this on an exam, I might not tell you anything. I might have a question about like an identifying factor of this protein is it's a multi-pass transmembrane protein. Is that right or wrong? A multi-pass transmembrane protein. Wrong. It is a cytoplasmic, a cytosolic protein. No. It's a protein that's likely secreted from the cell. No. It's a single-pass transmembrane protein. Yes. So it would be something like that where you'd have to use this. Yes?
So if I had more than one stop transfer sequence, would it be multi-pass?
Exactly. So the question was if it had more than one stop transfer sequence, would it be multi-pass? Yes. Yes. And I'm going to show you that in a minute. Yes.
Above the zero is hydro.
Yep. So positive, hydrophobic. Positive numbers are hydrophobic. Lots of hydrophobic amino acids are likely to form a transmembrane domain. A transmembrane domain is pretty much the same thing as a stop transfer sequence. Yeah.
How would you know if it's going to be sent to the cell membrane or just going to stay in the ER?
How would you know if it will be going to the cell membrane or if it's going to stay in the ER membrane? We will learn about that. The answer is that we'll have a K-del sequence, which is an ER retrieval sequence, which will basically make it not be able to. So if it ends up in a vesicle that goes outwards, there are these, there are ways for it to be brought back by recognition of signal sequences. They're not true signal sequences, they're ER retrieval sequences.
But you would be able to look at that.
You can look at a sequence and determine whether or not it's an ER resident protein. But we'll get to that. We'll get to it. Don't worry if you didn't get that because I'm going to teach it. Any other questions? Yes.
Did you say that this is a single pass or signal pass?
Single pass. One pass through the membrane. Any other questions? I know I think I spend too much time on this because people get so confused, but it might be that I spend time because people get confused. I don't know. Yeah.
Is there any specific way, or like, is it the amine group and the other group? Is it like one group stays inside the ER and the other stays inside the cytosol? Or
So the question is, how do you know which side goes in the ER and which side goes in the cytosol? It has to do with the sequence, and we're not going to worry about that. But if I show you it already in the ER membrane, and I ask you how it's going to look on the plasma membrane, then you should know, because always cytosolic stays cytosolic, and the ER lumen is similar or the equivalent of extracellular.
So which one is cytosolic?
In this case, I think we decided that the carboxy. And so that means it will be cytosolic when it gets to the plasma membrane, because it maintains the topology. Because a protein's not going to just pull itself out of the membrane and flip around, right? That'll be weird. Okay, let's keep going. Okay, so all this terminology, I have a little list. They're called topogenic sequences. These are sequences that have to do with the topology of a membrane protein. And so you might get a start transfer signal, which could be a signal sequence that acts as a transmembrane domain if it's internal. There are stop transfer sequences, which again are transmembrane domains. And they often will happen after an N-terminal signal sequence, kind of like what we just saw there. Stop transfer sequence after an N-terminal signal sequence. Can one sequence serve multiple functions? Yeah, sometimes you'll just have a signal sequence that's internal that will act as a transmembrane domain or stop transfer sequence. And so that's where it gets confusing. Just understand large 20 to 30 amino acids, hydrophobic, likely to be transmembrane. Can also be a signal sequence, right? Because you'll have to have some sort of signal sequence to target proteins to the ER that are going to be membrane proteins. Because that's the only way they'll be membrane proteins. They don't get into the membrane other ways. Okay? What determines the topology of the membrane protein? Which end is cytosolic, which end is luminal? It has to do with the number of start and stop transfer sequences. It has to do with the amino acids surrounding a stop transfer sequence. So don't get bogged down with those details because I'm not giving you sequences to characterize. Okay? But do understand that if what's cytosolic in the ER remains cytosolic on the membrane. Okay? Why is this important? Why am I telling you this? Uh-oh. I missed the mark. Yeah.
Protein, so the cell membrane cell communicates.
Right, so that these are going to be your cell surface receptors. These are the receptors that help the cell to communicate with the whole environment, with other cells. If this doesn't happen correctly, if a protein ends up upside down, then there's no communication, right? So it's important that we understand how proteins get into the cell, how they get into the plasma membrane, how they get in at the ER, and how they get up to the plasma membrane. Okay, any questions about this? I think I might have a few more pictures to show you. All right, here's a double pass transmembrane protein. So you would expect if you were, you know what I need to do is start using this thing. I never use it. You'd expect if you were looking at that plot, you'd expect that it would be something like, let's see, so there's our 0, and it's looking like you have your amino, and it's fine, and then you have this thing here, right? There's some hydrophobic, and then you might have something else, and then there's your hydrophobic, right? So that's your 1, 2, 3, and then your -1, -2, -3, right? So 2 transmembrane domains, you'd have 2 big hydrophobic peaks, right? And in this case, your start transfer sequence is acting as your signal sequence, and it's not cleaved, it's actually acting as a transmembrane domain. Here's your multi-pass transmembrane protein. So you actually would see 1, 2, 3, 4, 5, 6, 7 big hydrophobic peaks. So oh wait, I already asked you this. There's like 5 or 6 of you taking 410, and you have to do this with your small proteins, right? And look at the hydrophobicity. Okay, any questions about this? Okay, so this is the synthesis and insertion into the ER membrane of the GLUT1 glucose transporter. This is that protein that helps to bring glucose out of the blood into the cell. Right, so here you're having this internal signal sequence. So you've got translation, here's your amino terminus, and then there's your signal sequence. Your SRP recognizes it, brings it to the SRP receptor, it goes in, it's recognized, and it's actually acting also as an anchor sequence or a stop transfer sequence, or I guess in this case it could be start transfer sequence. And then you have another one, and so this is this multi-pass transmembrane protein. And so they move into the membrane, so those are 1, 2 transmembrane domains, and then you are getting more translation, and there's 3, 4 transmembrane domains, and they move into the membrane, and here's 5 and 6 transmembrane domains, and you end up with this 6 transmembrane domain protein with the amino anchor boxes terminus on the cytosolic side. Okay, so the part that's going to bind to the glucose is going to be over here on the luminal side, right? Because it's bringing glucose in from out of the cell. Okay, that's enough of that. So not only do we have membrane proteins that are translated right into the ER membrane, but we also can have formation of GPI anchored proteins. And this happens in the ER. And so you'll have your protein made with this transmembrane domain, just like we've been talking about, but it will actually have, there'll be a GPI anchor that's embedded inside the ER membrane, and it'll be transferred, the whole protein will be transferred to the GPI anchor. So it's attached to the plasma membrane, but by a GPI anchor rather than by a protein transmembrane domain. And so this is just this lipid-based anchor for tethering the protein to the plasma membrane. So do you guys see how this is going to be on the cell, tethered to the cell surface by this anchor? So I just got a request for a repeat. So you've got the GPI anchor bound to the protein, and this is in the ER lumen. But remember, the ER lumen becomes extracellular as the vesicle, which this will be in, will transport it up to the cell surface. Okay, so I just answered the question. What is the destination of GPI anchored proteins? Cell surface, yeah. Why GPI anchor versus just having a transmembrane domain? Has anyone ever thought about this late at night while watching Jay Leno? Is he still on? He's about to lose his job, isn't he? Or he's leaving, or I don't know. I don't keep up. Yes, go ahead.
Will it break and release?
So it can be strategically. So the comment was, can it break and release? It can be strategically released by activation of enzymes that will cleave it, phospholipases. Yeah, and actually trypanosomes, which are, who knows what trypanosomes are? Who loves parasitology and wishes there was a parasitology class here? Oh, come on. Yeah, we got one. Yay! One person wants a parasitology class. Okay, so we won't be having that class. So trypanosomes are a parasite, a eukaryotic parasite that cause African sleeping sickness. Anyone? Okay. Anyway, so they have a lot of their surface antigens are on their cell surface by GPI anchors, and they're really smart because they, as soon as the immune response begins to recognize those cell surface antigens, they release them. Awesome. Not for us, for them, right? Because if you want to evade the immune system, isn't the best thing to do get rid of all the antigens on your cell surface that make you recognizable by the immune system? Brilliant. Anyways, and then we do this too. Yeah. Why? So it could be strategic. You could strategically release the protein. That's just, the trypanosome is only one example. Okay? This happens in the ER, that's why we're talking about it. Okay, here's our summary of delivery of proteins to the ER. Targeting, right? You have to have that protein targeted to the ER, and how is it targeted to the ER? Yay! Signal sequence recognized by SRP, right? So that's what gets it to the ER. Then it needs to bind, so that's your SRP receptor, and then it's imported into the ER, and then it needs to fold and be modified and whatever, right? Everyone feel good about that? Yeah.
Up until the stop, when we start the transfer sequence, or the transmembrane domain, I guess, is the protein being translated into the lumen?
Yes. The question is, up to the transmembrane domain, is the protein being translated into the lumen? Yeah, so it's just like what we learned, where you have co-translational import of the protein into the ER, and then once that transmembrane domain is recognized, it can move over into the membrane of the ER.
So why wouldn't you be able to figure out which side is the cytosol?
Because it depends on which side, how it interacts with the side. It depends on how, because it seems like it's just goes straight in, right? And then your N-terminus should always be on the inside, right? But not really, because if it binds to the translocon kind of upside down, let's say you have a bit of translation, then your start transfer sequence, or I guess signal sequence / stop transfer sequence, and then it interacts kind of upside down-ish, where you have your N-terminus sticking out, and then it will keep translating, and then your Karbacci terminus will be on the inside. So that's why I'm saying let's not get too bogged down with this, but just understand that they both happen, and it all just depends on the job of that protein and where the cytoplasmic tail needs to be and where the extracellular domain needs to be. Yes?
Sometimes we depends on where the start sequence is, which transcription inside service.
It pretty much depends on where your start transfer sequence is, and also the context of the amino acids around it have something to do with how it's going to interact with the translocon. And I'm not going into that level of detail. Yes, and were you raising your hand or just a little dancing? Okay. Any other questions? Okay. Okay. Proteins in the ER. Aren't you guys glad we finally made it into the ER? Woo. Okay, so what happens once a protein's in the ER? You get all sorts of modifications that help with folding and all this good stuff. Disulfide bonds are formed. You have folding, which is part of disulfide bonds and folding together. Addition and processing of carbohydrates. Big chunks of sugars are added. This also happens in the Golgi. So it's not just in the ER. Things are added in the ER, and then more processing happens in the Golgi. And that difference between what the sugars look like in the ER and in the Golgi actually is useful for when people are studying where things are. You can actually you can differentiate between what the sugars look like and tell where that protein is. Proteolytic cleavage, there was a question about that where someone was asking about whether or not that has to do with cleavage of the signal sequence. Not cleavage of the signal sequence, but actually sometimes a protein will be made. So it's this immature form of the protein, and you don't want it to be active until it's cleaved in some way. And that cleavage can happen in the ER or the Golgi. And then assembly into multi-subunit proteins. So often you'll have multi-subunit proteins that again might need to be, well, it doesn't matter why, but yeah, that will also happen in the ER. So that's a lot for the ER to do, right? And a lot to go wrong also. Okay, so what was I just talking about? Right, so you've got your ribosome translating your protein into the ER, and then you have to have chaperones that will come and bind it and help it to fold, and then disulfide bonds will be made, and then there are specific enzymes that help to catalyze the rearrangement of disulfide bonds, Then there are enzymes, peptidylprolyl isomerases, help with rotating around peptide bonds to get the folding to work properly. All that needs to happen. And if it doesn't happen correctly, and it can't, so proteins cannot get out of the ER unless they're properly folded, and so they'll stay in the ER folding over and over and over and over again until eventually some signals basically prevent the protein from being retained in the ER, and then it's kicked out and degraded. So either you can get out because you're folded properly and go on and live your happy life, or you're kept in the ER to fold, and if folding doesn't work, then you're degraded. Okay? So someone asked last week about chaperones. Are there chaperones in the ER? There are. BIP is one of them. There's HSP70 family of chaperones. HSP70 chaperones are just 70 kilodalton chaperones that bind proteins as they come off the ribosome and help them to fold. They're cytosolic HSP70 chaperones. They're ER resident HSP70 chaperones. They're mitochondrial HSP70 chaperones. They're all just the same size, which is why it's 70 and HSP for each protein. So you can see here, it's binding to the protein as it's coming off the ribosome, and it's preventing the hydrophobic domains, any sorts of hydrophobic domains from binding to each other and aggregating, and allowing it to fold. Any questions? Protein disulfide isomerase, or PDI, catalyzes rearrangement of disulfide bonds. So disulfide bonds are kind of going to form randomly as protein comes off the ribosome. Whenever there are sulfhydryl groups that are close enough, because of the reducing environment in the ER. See, I want to say that all the time. The oxidizing environment in the ER, you'll get formation of sulfhydryl bonds. And what happens, so here you've got your polypeptide that just came off the ribosome, and they're usually going to form just in order of that they come off, because those are the ones that are going to be closest. But that's not necessarily right. So PDI will come along, and it will bind to the sulfur groups and help in rearrangement of the disulfide bonds until you get the correct folding. Okay, so it helps with rearrangement of disulfide bonds. And then the reducing environment in the cytosol is what normally prevents formation of disulfide bonds, and that's why you don't see that very much. Okay? Questions? So why disulfide bonds? What do these do? They're stabilizing protein structure, right? We talked about this. Pretty much just talked about this. Okay, so then there are peptidylprolyl isomerases. And what they do is they rotate amino acids around the peptide bond to help with folding. Okay, what else happens in the ER? So we've got disulfide bond formation, we've got folding because the chaperones are binding to the protein and they're rotating. We've got enzymes that are helping to rotate amino acids around peptide bonds. Then you also have glycosylation happening. And so there's this enzyme called oligosaccharyltransferase that's in the ER membrane, and well, that's not it. And what it does is it transfers these large oligosaccharide groups from the ER membrane. I feel like it's really boring in here because I keep seeing people leave there. Going. Should I do more dancing? I'm gonna have to get more excited. End of day, woo! It was just like, I always notice when people get up and leave. So like when it's like one person, I think, like, well, they must have an important phone call. Oh, did you hear from your dad? See, important phone call. I get that. Or potty break. Everyone has to. See, I don't get to go if I have to be. I have to stay here. Because if I left in the middle of lecture to go use the bathroom, you guys would be like, what is? You guys would think I was sick probably. Like really, like there's something wrong. But anyways, okay, back to work. So this whole entire sugar group is transferred to the asparagine on the growing polypeptide chain. And that's why it's called an N, like letter N, linked oligosaccharide, because oligosaccharide is linked to the asparagine, and the single letter code for asparagine is N. So why do we glycosylate? Most proteins that go out of the cell or on the cell surface are glycosylated. You ever notice that? Wandering around. So the N-linked oligosaccharides are added to most proteins in the ER. You can have other sugar groups added to proteins in the cytosol. It's not the only time sugar groups are added, but these N-linked oligosaccharides are added in the ER. Okay, let's take a moment to chat with our friends and do a practice problem. Therapeutic proteins, such as antibodies, tissue plasminogen activator, which is an anti-clotting factor, and erythropoietin, which stimulates production of red blood cells. They used to be mass-produced in bacteria, but it was really inefficient and expensive because the protein would not fold correctly and would aggregate. This problem was solved by changing the host to cultured mammalian cells. Why did this change fix the problem? Do you want to take a minute and chit chat with your friends? I'm feeling that we're needing a collective break. Okay, so let's take a minute or two. Talk to your friends about this.
Now we're talking about
Oh my God, I thought you meant like skeleton. I was like, why? Okay, guys, let's stop. Okay, I let that one go too long, I'm sorry. I went up there and I talked to the people and then, okay. So, why did this change fix the problem? Did everyone understand the question? Okay. Who that I never see can tell me why changing to mammalian cells made production of these proteins so much more efficient? The real question is, what's the difference between bacteria and mammalian cells in terms of protein folding? Right? Because let's see, antibodies, tissue plasminogen activator, and erythropoietin are all proteins that are secreted out of the cell. Yes? Yeah.
Doesn't it depend if it's
Does it depend on if folding is what?
post-translational or post-translational?
Does it depend on if protein folding is post-translational or post-translational? Well, these are all proteins. Okay, so most protein folding is going to be
Well, no, let's get some more information. Yeah, yeah,
Is it because the bacteria cells don't have those membrane organelles
you're getting there. Is it because bacteria don't have membrane-bound organelles?
Do they not have the resources to like take apart and like refold proteins?
Do they not have the resources to take apart and refold proteins? I think they have their own ways, because they have their own proteins. Yes.
They don't have an ER.
They don't have an ER. Bacteria do not have an ER. Yes. Is that what everyone was thinking? Yes. They don't have an ER, they can't make disulfide bonds, and so these are human proteins. So yes, bacterial proteins have their own ways of becoming what they are, but these are human proteins that need to be folded for human use, and they need disulfide bonds, and they need to be glycosylated, and they need whatever they need has to happen, and it can't be done by bacteria. Good job, folks. Okay. Anyone want to add to that? Did you want to add anything?
I want to ask, so they want to aggregate, so they're going to aggregate just like the hydrophilic parts of them. So would you say there's just chaperones, or the bacteria were like lacking chaperones?
So the comment was, well, you say they're aggregating. That means hydrophobic domains were interacting with each other. Does that mean that they didn't have chaperones? Well, you have to go back then and ask yourself, why were they aggregating? Because yes, chaperones are going to bind and help things to not aggravate. Probably if they aggregate, it's also aggravating. But yeah, they're going to help things to not aggregate, but they're never going to fold properly. So bacteria have a lot of the same chaperones that we have. They're slightly different, but they probably bind and work just fine. But without making those disulfide bonds, that's a big one. Or without being able to rotate around those peptide bonds to fold, then the chaperones can't do their job. Okay, any other thoughts? Okay, good. So these are the kind of questions that I like to put on exams in some kind of multiple choice format. I'm making you use information that I taught you, hopefully. That's the goal. Okay. Okay, so we've got proteins that are folding and sugar groups that are added. So calnexin and calreticulin are proteins that live in the ER, and they bind to sugars, these big sugar groups that we added with the glycosylation. And they retain the proteins in the ER until they're properly folded, right? Because we don't want folded, misfolded, or unfolded, or wrongly folded, or I don't know, not properly folded proteins leaving the ER. And so here it's showing your ribosome translating your protein in through your translocon, and these carbohydrate groups are being added. It's not really perfect, because if it was perfect, it would show that the carbohydrate groups were in the membrane and being added, but whatever. And then there's calnexin, which is this membrane-bound ER resident protein, which we talked about that there are membrane-bound ER resident proteins. This is one that binds to the carbohydrates and keeps the protein in the ER while it's folding. So there's this one that's bound in the membrane, and then calreticulin, which is in the lumen of the ER. They both bind to the carbohydrates and participate in folding, while all the things that disulfide bonds are made, all the things that need to happen in order for the protein to fold properly. And then this example is showing the folding of hemagglutinin precursor. Who knows what hemagglutinin is? HA. Someone must know. Hemagglutinin. Who had the flu shot? Well, so your bodies all know. No? Are you a flu shot hater? Sorry. I'm a vaccine lover, so whenever people do like this, I used to work in a lab during my postdoc, And this guy, another postdoc, and he was like a lifelong postdoc, he was an MD, didn't believe in vaccines. And every year he wouldn't, so this is at Hopkins where they're like, if you walk by, they like throw a vaccine at you. Like, it's awesome if you get them for free. And he did not believe in vaccines and told me every year that it gave him the flu. And I was like, how did you get your MD? Who gave it to you? And I want to take it away because there's no way. But hemagglutinin, they talk about H1N1 and all that. That's the H is for hemagglutinin, which is a receptor envelope protein for the influenza virus. And so it's made in the ER, right? Does the flu virus have an ER? What ER are we talking about? RER. They're hijacking RER for their proteins. And so it's a trimer. So remember we talked about assembly of multi-subunit proteins, and so this is an example of what's happening with HA. There are these precursors, and they fold, and then they're assembled into the trimer, and then you've got your viral protein in your ER. How dare it? But the whole point here is calnectin and calreticulin are keeping that protein in the ER until it's properly folded and assembled into its trimer. Any questions about this? I used to do a recitation paper on this, but I believe I removed it this year. Because, well, we need to change things, right? So only properly folded and assembled proteins can leave the ER. And what this is showing here is this is an antibody being assembled, and BIP, which is that ER resident chaperone, is binding to an antibody heavy chain, because antibodies are multi-subunit proteins. They're 2 heavy chains, 2 light chains. And so you've got your chaperones holding onto it. And then here it's not showing calnectin and calreticulin, but they are what are going to keep the protein in the ER until it's assembled into a full antibody, which then will be incorporated into a vesicle and will bud off of the ER, go to the Golgi, and then go to the plasma membrane and be secreted out of the cell. But only properly folded and assembled proteins can leave the ER. So proteins that cannot be folded are removed by ERAD, ER-associated degradation. And then there's also the unfolded protein response. So if there's one protein that there's a mutation in, like there's all sorts of proteins that get mutations and can't fold properly, then that will just be removed from the ER and degraded eventually. If there's some kind of more systemic issue, then it will trigger the unfolded protein response. So this is 2 things. One is ERAD, one is the unfolded protein response. Students tend to lump them together. So I'm telling you now, don't lump them together, they're 2 different things. ERAD is removal of proteins that cannot fold properly, likely due to mutation, and the unfolded protein response I'll talk about shortly, looking at the time. For a very short time, apparently. The unfolded protein response actually induces transcription and translation of proteins that assist in proper folding. So there's this massive amount of unfolded proteins that are sensed, and then you get activation of gene expression of genes that are associated with protein folding, such as chaperones, peptidyl-prolyl isomerase, protein disulfide isomerase. It's kind of more of like a heat shock response kind of response. So this is what it looks like. So this is one, I have a few slides that explain this, but the basics are that there are these sensors that are in the ER membrane, and they've got the luminal side that will sense unfolded proteins, and then they've got this kinase side that I've seen two versions of this. One kind of shows the kinase side in the nucleus, because remember, the ER membrane is connected to the nuclear envelope, right? And then we'll see in the next one, it doesn't show it so obviously in the nucleus. But either way, you get this activation of this kinase, which does multiple things to induce expression of genes that are associated with protein folding. But one of the things that it does is it activates a endoribonuclease. What's an endoribonuclease? Endoribonuclease. What is a nuclease? People are murmuring. I know there's an answer out there. Nuclease. What's a nuclease? Come on. Don't raise your hand. My kids are really into raising their hands right now. They just started kindergarten. Nuclease degrades what? Nucleic acid. Okay. Ribonuclease, RNA, degrades RNA. Endoribonuclease. Okay, so it's cleaving out a section of RNA. And it actually, what it's doing is it's splicing out an intron from a preformed RNA precursor. So you've got this unspliced RNA hanging out, and then the endonuclease cleaves that out, and then you get that the exons ligated together, and then you have then the mRNA is now spliced, and it can leave the nucleus, and it can go on and be translated to make this transcription factor called HAC1. Don't worry about the name. It's a transcription factor that will then go back into the nucleus and activate expression of genes such as genes encoding chaperones and different things that are needed for dealing with the fact that the ER is full of unfolded proteins like peptidyl, prolyl, isomerase, and all those good things. That's kind of cool, huh? No, a little bit. There's actually like a sensor that says, yo, I have unfolded protein here. And then there's this preformed RNA that just has one leftover intron that needs to be removed. So it's like it's ready to activate gene expression. So there's three main pathways actually that are activated by unfolded proteins. So one is this this splicing thing that I just talked to you about. There's this mRNA that's spliced that activates expression of these gene regulatory proteins that then goes back in and you induce gene expression to increase the protein folding abilities in the ER. And then there's another pathway that I didn't talk about, which, remember, these are kinases that get activated. What do kinases do? They phosphorylate things. What phosphorylation do usually allows proteins to interact, or something like that, or prevents proteins from interacting. So these kinases get activated, and then they inactivate translation, right? Would you want to inactivate translation? Maybe a little bit, right? So you might want to activate some translation of things that are going to help protein folding, but you might want to also tone down translation of proteins into the ER, because if you're having an unfolded protein response, you probably want to lighten the load a little. So slow things down a little bit so that unfolded protein response doesn't get worse, or unfolded protein burden doesn't get worse. And then, what was the other one? I'm forgetting what the other one is. But that's okay, because we've got 2 minutes left. So I'm going to let you guys go, and we will finish up on this one.